All right, good afternoon. My pleasure to welcome you to the Dale Bauman Lecture this year. My name is Tom Overton. I'm professor and chair of animal science, and this is an event that we're delighted to do every year in collaboration with food science and division of nutritional sciences as well. And so it's also great to have Dale here this year in attendance. So great to have you here, Dale, and thank you, Colleen, for helping to make that happen. So I'm going to introduce Ron Butler here in a moment, and he'll kind of get you oriented to the lecture. Just want to do a couple quick thank yous. So Shing and Lei, as well as Ron, are kind of the drivers on this lecture. So thank you for that effort. And then also Barb Jones providing support in lots of ways to make this happen as well. Okay. And so with that, I will make sure we're sharing the appropriate screen and ask Ron to come forward. All right. On, we should be good, I think.
And good afternoon to everybody. So the Dale Bauman Lecture was established to recognize the remarkable career of our friend and colleague, Professor Dale Bauman. And his research spread across animal biology, food science, and nutritional biochemistry. For your program, you already know some things about Dr. Bauman and his research. He did a lot of work with dairy cows, and on the front of your program, I know it's a small picture, but in the background, there is a real dairy cow. And Dr. Bauman really knew about dairy cows and probably still does know a lot. The dairy cow was Dale's primary research animal, and he did a lot of whole animal research, and through that whole animal approach, as well as studying various organs and tissues in the dairy cow, he recognized that there is a remarkable coordination and partitioning of nutrients during both pregnancy and lactation. And his research primarily, I think, brought new emphasis to an old physiology term, which I listed here, homeoresis, which means the orchestrated changes for priorities of a physiological state. Dr. Bauman still is actually one of the most highly cited authors in the agricultural sciences and other disciplines. I updated the numbers here just in the last couple days, and you can see them here. He gets citations all the time for his various work. And you saw his various awards. Here's just a quick example of the breadth and standing of Dr. Bauman that was recognized in this journal cover for his classic studies identifying the key enzymes regulating the synthesis of milk fatty acids and lactose during lactogenesis. A real diverse set of interest and background. Dr. Bauman was elected to the National Academy of Sciences, and he served as chairman of the board of Agriculture and Natural Resources for a number of years. Dr. Bauman has joined us today. Dale, would you kind of get recognized? Okay, I'm going to turn the program over to Julia Finkelstein, and we'll go from there.
Good afternoon, everyone. It is such an honor to welcome Dr. Shelley McGuire back to her alma mater here at Cornell University. I'm absolutely delighted. So welcome back, Shelley. Dr. Shelley McGuire, she is a DNS alum, so she graduated from the Division of Nutritional Sciences here at Cornell in 1994. She's also one of our NICHD T32 alums from our maternal and child nutrition training program here at Cornell. Shelley is professor of nutrition, and she is director and Margaret Ritchie School of Family and Consumer Sciences. And she is also director of the NIH Center of Biomedical Research Excellence in Nutrition and Women's Health at the University of Idaho. She earned her Bachelor of Science in Biology and her Master of Science in Nutritional Sciences from the University of Illinois at Urbana-Champaign. And of course, she earned her PhD in human nutrition just down the road in the Division of Nutritional Sciences with minors in physiology and, sorry, I need new glasses, sorry, with minors in international nutrition and physiology. Dr. McGuire has been fascinated with every facet of human milk and lactation, and she has devoted her career to understanding how maternal physiology, diet, and environment shape milk composition. Working closely alongside her husband and longtime scientific collaborator, Dr. Mark McGuire, she has uncovered key insights, including the links between maternal energy balance and milk fat, evidence that certain trans fatty acids may suppress milk fat in lean women, and associations between postpartum fat loss and shortened anovulation. More recently, her research has focused on the extraordinary variation in human milk composition, with emphasis on complex carbohydrates, the milk microbiome, including pathogens such as SARS-CoV-2, and the effects of maternal cannabis use. Dr. McGuire is a star in the field of nutrition. She was elected to the National Academy of Medicine in 2022. She was named a Distinguished Fellow of the American Society for Nutrition in 2025. And just on a personal note, I didn't have a chance to overlap with Dr. McGuire here at Cornell, but I did have an opportunity to see her as part of the Dannon Nutrition Leadership Institute. I was in the last cohort of early career scientists, and which of course Kathy Rasmussen and Shelley McGuire led for many, many years. And not only is she an extraordinary scientist, but she is an incredible human being and mentor. And I can tell you from a personal experience, she is an absolute incredible mentor in the field of nutrition and maternal and child health. And we are absolutely honored to welcome you back to Cornell. Please join me in welcoming Dr. Shelley McGuire.
That wasn't really me. Anyway, thank you so much. It's humbling to be here. I got to spend the day with Dale, so that was incredible. Thank you for that. Yeah, it's great to be back to Cornell. I haven't been back in a long time, and it was a great place to get trained. Thank you, Kathy, for all the training. And I think I'll just get started. I'm going to talk today. I'm going to try to tell a story about some of the work that we've done collaboratively over the last 30 years. Since we left Cornell. And it's a lot about asking simple questions that are not always easy to answer, but they are simple questions in many cases. And I'm just going to start by saying that I work with my husband. He's right here. This is our lovely family. These two beauties were born here in Ithaca, Emily and Grace, and then our son Keith was born in Idaho. So we've really done almost all of our work together. So everything I talk about today really has been done as a team. Most recently, I've learned to play the bagpipes and Mark is playing the drums. And that's us in the University of Idaho graduation just last spring. But really everything that we do, this is now going. Okay. Everything we do, we've done together. So I'm just going to put that right up front. So this is Mark's work as well. The other thing I want to mention before I move on is that mentors matter. And I was lucky enough to be mentored by Mary Frances Pachano at the University of Illinois, who brought me into the field of human milk and lactation, and I never turned around after that. Is that better for you?
I think so. Well, sorry.
Yeah, I don't think that is coming to the speakers.
Is this not in?
Why don't you go ahead and
Okay. And again, Kathy Rasmussen was my PhD advisor here. So I have a pretty good pedigree in human milk and lactation that really has come in handy. But I also want to point out other advisors and committee members that mentored me along the way. John Milner, who's up here on the left, Mary Frances's husband at University of Illinois, had what was called the tag team committee at University of Illinois. They were married and you always got them to argue with each other. And then you didn't really have to do anything because they just kept arguing. But also Dale Bauman was not on my committee, but I ate a lot of lunches in the storeroom in the animal science building and learned a lot from Dale. My committee members were Alan Bell, Ron Butler, and Jerry Haas, and I wasn't expecting Jerry to be here, so what a treat. And then Jean-Pierre Habicht was my sort of informal mentor on international nutrition as well, and I learned a lot from him, so I just wanted to mention that. Enjoy your mentors. They're lifelong mentors, actually. So I'm going to talk really quickly about just a few topics that we've looked at. It's kind of fun to put together a presentation like this because you get to pick and choose. And interestingly, as I was putting this talk together, I realized how much of this really was following up from Dale. I hadn't realized it so much till I put this presentation together. So when we left Cornell University, we started looking at milk fat. And I'm going to talk a little bit about that, conjugated linoleic acid and trans fatty acids. But then we moved into the milk microbiome because of that. It was a logical progression, which I'll tell you. We thought milk was sterile. Turns out it's not. And I will say that my ability to do this well actually was also supported by my work here, because this is when I started doing international nutrition research, which I learned a lot about here at Cornell. We did a little bit of really contentious work with pesticides with Monsanto. And again, there's a Dale connection with that because he had been working with Monsanto on BST for years. And then I'm going to talk just a little bit about our work with COVID, which felt like flying a jumbo jet while building it, and some of our current work looking at cannabis use during lactation. So I'm going to start by saying that none of this was a straight path, and none of this was planned. We really looked at research as what should we be doing? What is interesting? What's going to impact human health? What do we have money for? You know, and we've studied a lot of things in milk. And I want to point out too that this is sort of my professional track. It also wasn't very easy, to be honest. This is a whole other talk, but I started at the University of Idaho as a spousal accommodation in Mark's lab. I made $12,000 a year. It was great. And then I moved to Washington State University, where I was on the tenure track, but I was part-time. I was their only part-time tenure track position, and I did that for years. During that period of time, we had 3 kids. They were very busy. We didn't miss a soccer game. We went to every kids thing we could go to. This was not a simple path. At WSU, they closed the nutrition program, and I ended up going to 4 different departments in 4 different colleges. So you never know what's going to be ahead of you, but you just have to make the best of it. A few years ago, I went back to University of Idaho, where I'm now the director of the School of Family and Consumer Sciences. Here, that's called Human Ecology. And I'm pretty busy as an administrator, but we're still doing research. So these are the things I'm going to talk about. This is sort of the order that we've studied them. I'm not going to talk about postpartum fecundity. We did a little work in that. We've done quite a bit of work in genomics and mastitis, and I'm not going to talk about those either. So I'm just going to touch on these other areas of interest. Okay, so when we got to the University of Idaho, I had been studying, let's see, here with Kathy, regulation of postpartum and ovulation in a rat model, right? I hadn't really done a human study. And we were looking for the next best thing to do. And Mark was really interested in milk lipids. He'd worked with Dale on milk lipids. And we knew that total milk lipids were highly variable. Fatty acid concentrations were highly variable. We knew a lot about what was going on in the cows. Not so much about women. I really hadn't considered this much because I didn't even take, was it Dr. Zilversmith's lipids class? Because I thought, I'm never going to study lipids. But here we go. We started getting interested in lipids. And again, this is a recurring theme. People matter in your career. And what happened early in my career at the University of Idaho is we basically got, I think, an email or a phone call from this guy, Bob Jensen, who had written what we call the Blue Book, the Bible of milk composition. And I think did he email us or call us, Mark? I don't know. It was an email. It was an email. And he said, you guys should study conjugated linoleic acid and trans fatty acids in human milk. And I'm like, why? And he said, just trust me. It's really important and you should study it. And I thought, okay, why don't we do that? Because we're just getting started. So because of Bob Jensen's sort of cold call, and I respected him and trusted him, I knew him from the Lactation Society, we decided to do a simple, very, very simple study on conjugated linoleic acid in human milks. Now, is anybody in here not heard of conjugated linoleic acid? It's okay. You can raise your hand. That's fine. Right. So this is a fatty acid. This is actually a group of fatty acids 18 carbons, 2 double bonds, and the double bonds are conjugated, unlike linoleic acid where they're not conjugated. And there's various forms of them. And here are 2 forms, the trans-10 or the trans-10 cis-12 CLA and the cis-9 trans-11. Don't worry about those numbers and the placements of the bonds. It doesn't really matter. But what's important is that this was a pretty hot topic at the time. It turns out that CLA is high in beef and dairy fat. It's a potent anti-carcinogen, probably the most potent naturally occurring anti-carcinogen. Very, very interesting. We knew at the time that bovine milk intake could negatively be related to breast and colon cancer in humans. So people were wondering, is this a really healthy fatty acid for humans to consume? We originally thought that this these fatty acids were coming from rumen biohydrogenation. So coming from the rumen and then getting put in the milk or the fat of bovine animals. In Dale's lab and our lab, we also later learned that it's also made in the mammary gland, but at the time we thought it was made in the rumen. And so we asked a very, very simple question, probably the simplest question ever. Can we detect CLA in human milk? And can we detect it in formula? Seems like a very simple question, and it actually was. But I want to say here, you know, this was a stage of our career where people were saying, you need an independent research program. We never worked independently. Interdependence is way more powerful than independence. And that's a concept that I'm going to come back to frequently. So this was our first interdependent study that we did at the University of Idaho. Very simple, of course. We collected human milk. We collected 4 different brands of formula, 2 different lot numbers of each kind. We looked at powdered versus liquid, you know, very, very simple study. The most simple thing we could do. We collected milk from 14 healthy women by complete breast expression. I had learned here that methods matter and you collect your milk right. And then, of course, we validated the assay for human milk, and it had already been validated for bovine milk. So very simple, very simple. To sort of go through some of the results, these are the data. I'll just explain it. These are the data from the formula. It turns out that there was no CLA in most of the formulas, and the only formulas that contained CLA were made with beef tallow. Not a big surprise, right? The ones that were made with vegetable oils had no CLA. And it really didn't matter what form, if it was powdered or ready to eat. And then what we found, here we have the human milk values. Let's see if I can get this to work. Yeah, it doesn't work very well. Human milk here's on your left, infant formula is on the right. These are the 2 forms of CLA that I showed you before, the trans-10 cis-12 in the black, and then the more naturally occurring form, the cis-9 trans-11. You can see that there's a lot in human milk, okay? Now that was interesting to us because these were at the same levels then compared to what we were seeing in bovine milk. And we were surprised that there was that much CLA in human milk, right? Because we thought it was being made in the rumen. I really expected lower amounts. So we weren't surprised it was there, but we were surprised at how high it was, and it was also quite variable. So the question was, where was it coming from? Was it just coming from diet, or where was it coming from? And so we moved on to ask another simple question, which is basically, if a woman consumes more CLA, does the CLA in her milk go up? And we had to attack this from a few angles. At the time, we couldn't get single isomer CLA supplements. All we could get were these mixed CLA isomers. And we did some very simple studies using both supplements and then food. And I'm going to go through a few of these studies that we did. So this was a really, really simple study. We asked if women consumed a CLA supplement which contained mixed isomers, does milk CLA increase? Again, very simple question, very simple question. But we did the study because we needed the answer to it. This is just to show you the CLA isomers and the different fatty acids that were in. We had a placebo and we had a CLA supplement. And you can see here that basically the placebo didn't have any of the CLA isomers, but the CLA supplement did. Of course, what you'd expect. And then when we looked at the milk fatty acids, here's the CLA treatment, the washout treatment, and the placebo treatment. We like to do crossover studies. And you can see here that the cis9 trans 11 form was the highest, of course, when we gave the CLA supplement. That's what we hypothesized, and that's what we found. So that wasn't a big surprise. What was a surprise, however, is that we had collected these samples because we were looking at milk fat. So we collected complete breast expressions, standardized for everything, because milk fat is highly variable and you have to collect your milk right to really look at it. And what we found here was a surprise, and that is this is milk fat content during the placebo, the washout, and the CLA period. And you can see that during the CLA period, we had a significant drop in milk fat. Now, milk fat provides the majority of calories to babies, so this was concerning. And these supplements were on the market. So we were a little bit concerned about what was going on here with this, what looked like milk fat depression. So we went on, and I'll back up. You saw in the slide about Dale that he'd done a lot of work on regulation of lipids. So we had this background, we had Dale's research, and we were very interested in understanding what was going on with milk fat. So at the same time we were doing the supplement trial, we were also looking at CLA consumption from foods, and we ran a few studies which kind of cross over with the food science whole world, which I think we like to do. We like to actually look at real foods and how that affects human health. And one of the first studies we did in this regard, we called the ice cream study, because basically we enrolled people, they love this study, we enrolled women, we said, what kind of high fat dairy products do you like? And then we delivered bags of it to their homes. And we said, eat as much of this as you possibly can. And then during the washout periods or the control periods, we gave them other high fat foods that did not contain CLA. So again, we had a depletion period, we had a high dairy period, and we had a low dairy period. And what we were trying to do is see how high we could push that milk CLA. So here you can see on this table, this is just nutrient intakes during the, you can see here during the high dairy period, they were consuming a lot more CLA than the other periods. So that was our dietary intervention group or intervention treatment. And here are the milk fatty acids. And you can see here is the depletion period, the high dairy period, and the low dairy period. If you just look at the top, we did increase milk CLA, but it didn't increase a whole lot. We were quite surprised because if you look here, we'd increased CLA intake from 34 to 291 milligrams a day. And here we didn't even double milk CLA. So we were getting really interested in what's going on with milk CLA. So again, we saw that a lot lower increase than we would have expected. And major problem with the study, of course, is that a lot of other things changed in the diet, right? We just gave them as much CLA as possible, but that also meant that they consumed a lot more fat. They consumed a lot more of other dietary components. So what was also interesting about this study is that, of course, we collected our milk the appropriate way to look at milk fat, because we were now interested in milk fat. And here's what happened to total milk fat. During the high dairy period, we had significantly higher milk fat. Now, you remember in the last study, when we gave the CLA supplement, we had milk fat depression. Here in the dietary period where we had more CLA, we actually have higher milk fat. So this was getting to be kind of interesting. What is going on here? And so the question we asked ourselves was it something that they were consuming during the dairy period that was increasing milk fat? Or was there something that they were consuming during the low dairy period that was depressing milk fat? And our hypothesis was that it was the latter. At this point, work coming out of Dale's lab and other labs was showing that consumption of some trans fatty acids decreases milk fat in the cow. So you can imagine in the study that we had just done, we were delivering them ice cream and butter during the dairy period. During the non-dairy period, they were getting a lot of margarine. Okay, and we were asking them to replace the butter with margarine. Now, this is when there was a lot of industrially produced trans fats in the diet. It's before the trans fats were banned. And so our hypothesis actually was that those trans fats in the dairy substitutes, if you want to call them that, were actually causing milk fat depression. So at the same time, I'm going to get back to that, but this is a good animal science, food science, human nutrition project, because at the same time we decided, let's make a functional food. Let's make a functional cheese. We wanted to have a dietary intervention that was isocaloric, isonitrogenous, et cetera, but contain high and low CLA. So we actually, Mark, worked with the dairy. They fed cows differently. I don't know what he did, but they made milk with high CLA or they made milk with low CLA. And we had the dairy at Washington State University. They make this incredible cheese in a can called Cougar Gold. It's really good. But they made high and low CLA cheese. And we used that as a dietary intervention one time. And so I'm not going to go through this whole table, but these are milk fatty acids of women consuming a regular diet. So it's kind of a baseline diet. And then one where we had them consume a standard amount of low CLA cheese and one that they consumed high CLA cheese. Okay. Now, I'm not going to go through all the details, but as you might expect, we were able to increase the CLA content of the milk when they consume the high CLA cheese versus the low CLA cheese. And this was a more powerful study because everything else was pretty controlled because the cheeses contained the same amount of other components. So that didn't really surprise us. And again, we were doing a lot of these studies at the same time. And during this period of time, there was a lot of research coming out on, what is the anti-cancer effect of CLA? And it looked like CLA was actually anti-inflammatory. So we got very interested in whether CLA could be anti-inflammatory in the mammary gland, in the lactating woman, et cetera. We'll come back to that. One last study I want to show you called the muffin study. You can see a theme here. We like to feed real people real food. And so at the same time, when we were interested in this idea of trans fats and CLA, we ran a study called the muffin study, which was more controlled than the ice cream study. Because what we did, I had this great graduate student, and she developed these recipes for extremely high fat muffins. And one muffin contained butter, and the other muffin contained regular margarine, just full of trans fats, right? So we were able to put women on these two different diets, isocaloric, isonitrogenous, iso everything, just different in butter versus margarine. And that was the power of this study, is that everything else was controlled. The butter group, we're not going to go through the fatty acids, this was just dietary intake, but the low trans margarine did contain very low amounts of trans, where the regular margarine at the time contained high amounts of trans fats. And of course, the butter contained CLA and the margarines did not. Now, we were able to shift fatty acids. That was at this point not that exciting. What was more exciting was what happened to milk fat, because our hypothesis here was that those high trans muffins were going to drop milk fat. And at this point, we'd also been doing quite a bit of work with body composition. So we did DEXs on our women. So we were able to look at maternal body fat. And in this table, I highlighted the parts that I want you to look at. What we looked at was milk fat in the butter group, the low trans margarine, and the regular trans margarine based on the woman's body fat. And it turns out that indeed, we were able to drop milk fat when the women consumed that high trans diet, but it was only in the lean women. So the lean women dropped their milk fat. And if you looked at NEFA concentration, so this is not a stereotypied fatty acid, free fatty acids, it looked like the women with higher fat were able to mobilize more body fat when they were consuming the trans fats. And somehow that probably kept their milk fat up to where it had been before, whereas the lean women were not able to mobilize that body fat. That's what it looked like. So these are pretty big differences in milk fat. These were not long-term studies. We never did long-term studies. These were pretty short-term studies, but that's a significant drop in milk fat, and it's of concern. So these findings really mirrored what Dale and others had been finding in cows. They made perfect sense. These industrially produced trans fats could drop milk fat in breastfeeding women just like cows. And this paper and several of these papers were cited in the decision that the federal government made to ban basically synthetic trans fats from the diet. Not the only reason, but a reason. And so, we can't do these studies anymore because we can't buy this kind of margarine. So anyway, we did a lot of other CLA studies, but I just want to come back to one more. We were always interested in why the CLA content of milk was so high. And this is a study, actually, I wasn't even a co-author on it. So this was an independent study by Mark. One of his grad students actually, oh, I am a co-author on this. There's another one that I'm not. But we use stable isotopes in lactating women, and we're able to show that women are able to synthesize cis-9 trans-11 CLA in the mammary gland from precursor fatty acid called racemic acid. And again, this mirrored data that Dale had from the cows. So this was just done in 4 women. We gave them stable isotopes of racemic acid, and they were able to produce CLA from that. So we now know that it's not only coming from the diet, but it's being made in the mammary gland, which is interesting in itself. Mammary gland doesn't usually put time and effort into things that aren't important. So still very interested in this fatty acid. And if anybody in this room wants a good career goal, I don't think anybody's done any more work on CLA in human lactation since we stopped looking at it a few years ago. So it's a very interesting area. So I'm going to move on and talk a little bit about the milk microbiome, which is probably what our lab is most famous for over the years. And this followed up directly from the CLA work, and I'll explain that. And I will say that when we started this research, we thought milk was sterile unless it was being produced by an infected gland or it was contaminated in the milk collection process. So that was the state of the science at the time. So why this paradigm shift? Okay, I want to ask you something. How many people see an ugly hag here, like an ugly old woman? How many people see a beautiful young woman? Okay, for those of you who saw one or the other, can you switch the way you look at it and see something else? They're both there, right? That's a paradigm shift. That's when you see something different because you've looked at it differently. Something made you look at it differently. And now you see something different. And this was a real paradigm shift when we realized that human milk is not sterile. So how did we do this? Well, this was the study that I wasn't on. So at the same time we were doing the CLA work, and I told you we were interested in inflammation, and there was some evidence that maybe CLA was anti-inflammatory. Mark got interested in that because he was doing some mastitis work, and he was working with some microbiologists, and we know that fatty acids are biologically active. And so he did a study where they treated Staph aureus and some other mastitis causing bacteria with a variety of fatty acids to see if those fatty acids could decrease the growth of Staph aureus. Okay, therefore decreasing inflammation or mastitis in the cow. And it's a nice paper, a lot of fatty acids. I'm just going to show you one figure. These are the data from CLA. So this is basically the growth of Staph aureus over time when treated with different levels of CLA. And the highest dose of CLA is here on the bottom. You can see, and the lowest doses are on the top. So when you treated Staph aureus with higher doses of CLA, you decrease the growth. You decrease the time it took for the Staph aureus to grow. That was really interesting, right? So here we have a fatty acid being made in the mammary gland. It's probably doing something there, probably important for the infant. And here it is. It was actually one of the most potent it decreased the growth, the most potent of any fatty acid. So we're very interested in that. And so we asked another simple question: is CLA consumption related to mammary inflammation or mastitis in women? Right? We have these supplements, like, let's see if it makes a difference to mammary inflammation in women. So we ran a study. We ran a randomized crossover trial, very controlled, with breastfeeding women, and we gave them, at this point, we had purified trans 10 cis 11 or cis9 trans 11 CLA supplements, and there was a placebo group. And we studied 16 healthy women. We took 3 samples during each of these time periods. Of course, we collected them in the right way because we're always interested in milk fat. And we had a lot of questions, but one of the questions we had was if the woman consumed CLA, maybe she would be less likely to have Staph aureus in her milk. Okay, so Staph aureus also causes mastitis in women, and you do expect to see it in milk if she's got some sort of inflammation caused by Staph aureus. So we sent the milk samples to the mastitis lab at Washington State University, and they came back and they said, all your milk samples are sterile. Okay, fine. I mean, we're looking at a lot of other things. But what was interesting is that we were also quantifying the immune cells in milk. And at the time, the best way to do that was to actually stain the milk and look at it under the microscope. So as we looked at the milk under the microscope, we saw a lot of bacteria, like in all the milk samples. And so here the mastitis lab is telling us your milk is sterile. We're looking at it with our eyeballs saying it's not sterile. And we're kind of interested in this. And I was giving a presentation at the University of Idaho in their biology seminar, and I presented this just kind of as a point of interest. And one of the audience participants, Larry Forney, who was participating in the Human Microbiome Project at the time, raised his hand and said, Shelley, maybe you have commensal bacteria in your milk. And I said, I don't know what commensal means. So I'm gonna have to look that up and get back to you. But he opened up this idea, let's look at this differently. He was working with people who were using next-gen sequencing techniques very early on with the human milk or with other microbiomes. And he helped us connect with them. And we were able to send them all of our milk samples. This was not something we could do in the lab at the time. And that resulted in a paper that's really considered the first report using these molecular techniques where we're looking at the microbiome of human milk. So again, this is a paradigm shift because we were looking at things differently. Just because something doesn't grow on a petri dish doesn't mean it's not there, right? We all know that from the microbiome world. So it turns out that all the milk, every single sample, and I'll go through that stack bar chart, every single sample had a really rich microbiome. And what you see here, this is each subject. So there are 16 subjects and each subject has 3 milk samples shown here. And you just look at the patterns, the color patterns, and each color is a different bacterial, I guess this is genus. No, yes, that's genus. And so a couple things to point out here is there's a lot of variation amongst women. And we see that with almost every milk component. There's a lot of variation amongst women. But within a woman, she had a pattern, kind of a fingerprint of bacteria. And so this was the first demonstration of a milk microbiome. These women were all healthy. There were no concern here. Interestingly, the top bacteria was Streptococcus and Staph. Again, nothing to be concerned about. Women were healthy. So this paper really launched a new scientific area of milk microbiome. This may be the only milk component where we saw it first in human milk, and then we looked at it in cow's milk. So yay, human milk research won this time. And I'll tell you, for those of the young scientists in here, we had a hard time getting this paper published. I thought, this should go in Science or something. This is cool. I mean, I think we submitted it to 5 journals because the reviewers came back and said, you obviously don't know how to collect a milk sample. You've contaminated them all. And we're like, no, I don't think we did. But we had trouble getting this published, and now it's a pretty highly cited paper. So don't give up when you're trying to get a good paper published. We had to, and I still stand in meetings and have to defend the possibility of there not being a milk microbiome. It's very interesting. So at that point, very, very quickly, other researchers started looking at this around the world. We had collaborators in Finland and Spain, and they're all pulling milk samples out of the freezer and analyzing them for milk microbiome. And we were finding different microbiomes in many cases. A lot of times the core bacteria would be the same, but the profiles were quite different. And there were a lot of things that could explain that. Either that could be real variation around the world, or it could be due to milk collection differences or milk analysis. There were a lot of things that could be causing those differences. So we became interested in that. And if there was that much variation, we were interested in what was causing that. And then, of course, the real question is, does that variation matter? Does the milk microbiome even matter to mammary health or to infant health? So These are not simple questions. These are complex questions, and they're questions that we've just continued to look at with collaborators around the world. And I just want to talk about a couple studies that we've done in this area. This was the first study that we had funded in this area to look at milk microbiome. The Gates Foundation funded this. And we did a longitudinal repeated measures study with 21 healthy women. We're always interested in healthy before we're interested in unhealthy, because we figure if you don't understand healthy, how are you going to understand unhealthy? So we had 21 healthy women. They collected milk samples at 9 times from day 2 to 6 months postpartum. Great subjects. They also gave us infant saliva, infant feces, maternal saliva, and maternal feces because we were interested in comparing microbiomes. And we had a few objectives here. The first was just to characterize the milk microbiome over the first 6 months. We did that. We found that it actually doesn't change a whole lot. I'm not going to present those data. It's quite stable from early on. And then, of course, I wanted to look at maternal diet and milk microbiome. I always think that maternal diet's going to be the answer to everything. So I wanted to see if maternal diet was related to variation in milk microbiome. And then we wanted to compare and contrast those different microbiomes. So I'm just going to show you a couple slides from this. This is a heat map. I think this is our first heat map we ever produced. We thought this was so cool at the time. And you had to actually drop in these figures like by hand on PowerPoint. Remember that? Couldn't get R to do anything. There was no R, I don't think. But this is a heat map. So we're looking at just correlations between different types of bacteria, different taxa. And then in this case, we're looking at energy yielding nutrients. And without going into the details, I just want to point out that there's a pattern here. And so you can see here we have a kind of a red stripe, and here we have a blue stripe. So red means a positive correlation, blue means a negative correlation. And so what it looked like here is that women who were consuming more energy yielding nutrients had milk that tended to have higher levels of firmicutes and lower levels of Bacteroides. We also looked at amino acids. This was kind of interesting. So this is amino acid intake and milk microbiome. Obvious patterns here. I don't understand this, but there's something going on with amino acid profiles, probably having to do with complete protein, animal protein versus vegetable protein. I'm not sure, but we have not followed up on this. I don't think anybody has. And interestingly, we also looked at micronutrients, and some of the strongest correlations we found with different microbes in milk, they look like nutrients from dairy foods. So vitamin D and biotin and iodine. So we've actually followed up a little bit on that, and I'm not going to go into it. So we do think that actually maternal dairy consumption might be impacting the milk microbiome that a woman produces. So those are some of our first studies. Have no idea if these same relationships will be found in other populations, and obviously we need to do some intervention studies to look at that. Okay, and the last slide on the milk microbiome is we compared complex profiles of microbiome in milk, so infant saliva, maternal saliva, infant feces, and maternal feces. And you can see that milk the composition is actually closest to infant saliva and maternal saliva. It's quite different from infant feces and maternal feces. However, that's the actual composition. If you look at just variation, let me back up here. Variation, so if you forget who's there, but you just look at variation in who's there, there's a really strong relationship between infant stool microbiome and milk microbiome. So even though they have different microbiomes, the variation is highly correlated, which might have impacts on infant health. We don't know. So some not so simple questions. Can we optimize an infant's microbiome by altering mother's milk microbiome? We don't know, but this is actually the state of the science right now. So I want to talk about one more study we did in this milk microbiome world. We're interested in all of these things that we think might be related to variation in milk microbiome, but I want to focus on geographic location because I consider this one of the most fun studies we've ever done. And so the question here is milk microbiome actually similar or is it different around the globe? And I had mentioned this earlier. A lot of people jumped into this field. We were finding different things. And we were just interested in whether those differences were real or were they methods related or whatever. And so to do this, I call this the Friends and Family Study. It was funded by the National Science Foundation. It was not a big grant. This was like our first big grant. And it was $1 million, which now seems like a lot. It seemed like a lot at the time. But when you see what we did, you'll see that $1 million actually, we were very efficient. Doge would have loved us back in the day. So this is a Friends and Family Study. This is sort of the main team that worked on this. And basically, I got a hold of all of our friends, colleagues around the world that I thought, hey, they know how to collect a milk sample. I want to work with them. And we invited them to be part of the study. So when you ask, like, why Sweden? I'll say, because Linda Kvist was there. You know, why Spain? Because Juan Miguel was there. So we did this study as efficiently and quite effective as possible. So again, Friends and Family Study. And one thing I want to point out is the relationships that you have with the people in this room and other people that you work with are going to stay with you the rest of your lives. Don't burn bridges, and you know, find out the people you want to work with and then work with them, because it makes work a lot more fun. Okay, so basics of this study, lots of populations, 2 in Ethiopia, 2 in Gambia, Ghana, Kenya, Peru, Spain, Sweden, 1 in US, California. These were Hispanic women because we were also interested in genetics. And then 1 in the Washington, Idaho area. We recruited about 40 moms and babies in each of these locations. We put together all of the collection kits in Moscow, Pullman, shipped them out all over the world. So everybody was using the same thing down to the glove, the collection kit, the diaper. We also collected infant feces. We sent these out. People collected the samples and they sent them back to us. We collected all the samples between 1 and 3 months postpartum. We ended up with 413 relatively healthy moms and babies around the world. And then we analyzed all the samples in our lab. So we tried to control for all of those other things that we thought might have impacted the milk microbiome, so we could see if these differences were real. I'm just going to show you one figure from the first paper that we published. Actually, I think this was the second paper we published from this. These are microbiomes in the milk that we collected around the world. Okay, so again, you're looking at stacked bar charts. You're just looking at color patterns. Each color is a different bacterial taxon. And these are our populations: rural and urban Ethiopia, Gambia, Ghana, Kenya, Peru, Spain, Sweden, and then the 2 US California, or the 2 US populations. And you can see there's a lot of variation around the world. This is what we had hypothesized, but now we were pretty confident that that was true, that variation was not due to milk collection or milk analysis. And just want to point out a couple things. In rural Ethiopia, one of the main bacterial taxa was rhizobium. We think this is coming from food fermented in the ground. This tends to be a soil bacterium, and it gets in the milk. Interestingly, in Ghana, there's a lot of Klebsiella. These women were not sick, so these are commensal bacteria. There's not a problem. They also had a lot of lactobacillus. They eat a lot of fermented dairy foods. So what we learned from this was a lot. We've actually published a lot of papers on this, and we've analyzed those milk samples for pretty much everything we can analyze it for. And we also genotyped the women, so we've done quite a bit of GWAS analyses, which I'm not going to show you. So really cool study, and it was a lot of fun because we got to work with people that we liked. So I have 3 last topics. They're going to be short. Just again, to kind of cover what we've been doing. We did a little work with pesticides in the middle of the milk microbiome work. Dale prepared us for this because Dale had worked for years on VST with Monsanto and got a lot of flack for that sometimes. Dale told me yesterday that you got a lot of emails from very upset people saying you were hurting children and things like that, right? So just to warn you, if you get into anything that's controversial like this, be prepared. I didn't realize how controversial this was going to be, but it was. But anyway, thank you, Dale, for sort of giving us a look into what might happen if you did this sort of work. So here's the story here. This woman Zen Honeycutt, who is not a scientist at all, knows nothing about scientists. She runs Moms Across America, highly reputable group, right? And she's one of these anti-Monsanto, anti-glyphosate, anti-Roundup, anti-all that, anti-tech. And she would do anything to shut down Monsanto, right? So she did a study where somehow they got milk samples from women around the country, and this is a slide from her website at the time. And they got the milk samples and they sent them to some lab who analyzed them for glyphosate, which is the active compound in Roundup. And they discovered really high levels of Roundup in milk. And this was all over the internet at the time. And there were, scary things like this, right? So you're toxic hazard, don't feed your baby. Of course, who's going to sit out in a field and whatever. But this was getting a lot of traction, right? So a lot of people were writing to the EPA and the whatever, and the FDA and saying, we got to shut this down. This is horrible. Monsanto needs to go out of business. They're killing babies. And I'm laughing now, but it wasn't funny at the time. And so we got reached out to and we're like, what do you know about glyphosate in milk? And I said, I don't know anything about glyphosate in milk. And I asked Mark, like, what do you know? And he's like, I don't know. Let's contact our friends at Monsanto, whom we knew through Dale. And they said, well, actually, we don't know anything about glyphosate in milk either. And we're like, oh, we should find out about glyphosate in milk. We should see if it's there, right? Because again, when Zen Honeycutt did her study, we knew nothing about how she collected the milk or how she analyzed the milk. We had every reason to believe that the analyses were flawed, that they were probably false positives. And so we're like, okay, well, we can answer this question, simple question. Does glyphosate, the active ingredient in Roundup, show up in human milk? Simple question. And actually, this was probably the simplest study we've ever done because we had just collected those 40 milk samples from our Inspire moms. They were in the freezer. We had collected them in a perfect way to analyze them for glyphosate. And so we're like, okay, well, we can do this study. So we actually teamed up with the folks at Monsanto, the devil, right? The devil. And because they didn't have, none of us had an assay that was validated for human milk. They actually spent a good year validating an assay for glyphosate in human milk. It was not easy. Human milk is hard to analyze. So they validated an assay in-house. We published that assay. They then transferred the assay to a third party, and we sent our milk samples to that third party for analysis. Critical aspects of this study, again, we collaborated with the experts. Okay, so for people who tell you don't collaborate with industry, I'd like you to think about that. Sometimes you actually should be collaborating with industry, with the people, and they had everything to lose if we had detected glyphosate in milk, and we would have published that, and they knew that. So we worked with them, validated the assay, we collected and stored the milk appropriately. We used a third party. We knew that we might, get some pushback. And so we did everything right. And I don't need to go into detail. We could not detect any glyphosate in any sample. It was below the detection limit. There's a metabolite of glyphosate. We also could not detect that in milk. We had urine samples, so we looked at those. We could detect glyphosate and AMPA in the urine sample. So we know that these women, we live in rural Idaho, they were exposed to glyphosate, but it wasn't showing up in their milk. So super simple question, actually pretty simple study. And I will tell you the harassment that came from this group after this was spectacular. I mean, threats, kind of death threats. I was FOIA'd. It took 2 years to get through the FOIA. It was really tough. So if you're going to do this sort of thing, which I highly recommend, because this is what we should be doing in land grant universities, just be prepared and make sure your university is going to support you in that. It was a mess. It was just a mess. Okay, couple last things. These are going to be quick. COVID, going to talk a little bit about that, and cannabis, the 2 C's. So to talk about COVID, I'm going to set the stage so you can remember how quickly things were happening when COVID hit, right? So here's a timeline of what we experienced. February 28th, first deaths in the United States. I was in Washington. We actually happened to be in Seattle that day when it happened. March 1st, we reached out to the WHO because we're like, you know what? This is a virus, just like HIV. HIV can get into milk. And we knew from experience that when we found out HIV could get into milk when the mom was infected, that was an international crisis, because then the question was, should infected women be breastfeeding their babies? And we thought, oh, let's avoid this, if at all possible, with COVID. So we reached out to the WHO, Larry Grummer-Strachan, and he was like, don't worry about it. It's not going to be a problem. Don't worry about it. We're going to be fine. I'm like, okay. I didn't believe that, to be honest. 10 days later, WHO declared COVID-19 a pandemic. A week later, we started galvanizing US researchers that were doing research that we thought would be interested in solving this problem, because I didn't think it was going to be business as usual. So we started that March 20th. That's actually a picture of me at the dining room table at our first COVID meeting. I think that's the first time I used Zoom, maybe. March 30th, the WHO came out with their recommendation, which is basically business as usual. Mom is infected, she should breastfeed, no problem. Well, April 1st, we started doing a lit search to see what we knew about SARS-CoV-2 and or any coronavirus. We'll talk about that on the next slide. April 2nd, AAP, American Academy of Pediatrics, and then followed suit by CDC, came out and said, if the mom is infected, she should be separated from her baby. So we're like, yeah, okay, we're not going to ignore this. So it wasn't business as usual. And this was happening in the United States. And anybody working with a breastfeeding mom at this time knows how stressful this was. So simple question 7, can SARS-CoV-2 be transmitted from moms to infants during breastfeeding? I think this is probably the most important thing we've done. Hopefully we won't have another pandemic and we won't have to do it again. But at the time, we were absolutely confident we could answer this question with colleagues around the country if we had the resources to do so. And again, we took this friends and family approach and reached out to colleagues that we've been working with for years to try to answer this. This felt like building a jumbo jet while flying it, because like we didn't have IRB approval, we didn't have an assay, we didn't have money, we didn't have this, but we just moved forward because we knew this had to be answered. So the first thing we did is we wrote this review. Remember I said we started it on April 1st. We submitted it on April 7th. There weren't many studies on coronavirus, or almost nothing on SARS-CoV-2. At the time we started writing this, though, there were 9 publications that had come out looking at infected women and looking at their milk. There was a total of 16 women in these studies, 20 milk samples. They had been analyzed using assays validated for plasma. They were all negative. There was no information on sample collection or anything like, did you use whole milk? Did you use a cell pellet? Did you use this? Nothing. So didn't really quite, it just wasn't good enough data. By the time this study was actually published, which was really quick, this got into literature really quick, there were 25 publications with 40 women, 80 milk samples, and 3 positives. Okay, so 3 of these milk samples were positive for the virus, and these were being published in like the Lancet. So now there was real concern because we were telling moms to be separated from their babies, and now we had published literature suggesting that in fact the virus could be in milk. Maybe it was, I don't know, but again, almost no information on how the milk was collected or analyzed. So we quickly got money from NSF and the Bill and Melinda Gates Foundation, and we, with our friends and family, we had no cases of COVID in Moscow, so we had to do this as a national study. We conducted a prospective longitudinal repeated measure study with 64 moms and babies. Moms were infected. Adults recently diagnosed with COVID, usually within the last week. So if you can imagine the logistics of this, we're putting together all the collection kits in Moscow Pullman so that we know they're clean. We're all in PPE. We're shipping these out to women who just told us they got COVID, asking them to collect milk samples in their home and shipping them back. The logistics of this were pretty spectacular, to be honest. We collected milk. We also swabbed the breast before and after. They cleaned the breast to see if the virus was on the breast. And all of this was done using standardized protocols and standardized materials that we put together in the lab, which made us very confident in the results. We also validated the CDC protocol for human milk, which was actually not that easy. Again, human milk is tough. And probably the most stressful part is I think every Friday afternoon we had a Zoom with Bill and Melinda Gates Foundation, somebody from WHO and somebody from CDC because they wanted to know the data. And most of the time we're like, I don't know. But anyway, when the data started coming in, it was nice. So we've published a lot of papers on this. The first one we put out with the first 18 women. There were 37 milk samples here that were tested for the virus. Going to detect the virus in any of the milk samples. Interestingly, on some of the breast swabs collected before we cleaned the breast, there was evidence of the virus. So these women had COVID. They were coughing. They were, they had symptoms, and there was virus on their milk or on their breast, but after they cleaned the breast, there was no evidence of that. What was really important here is none of the babies were sick. So even though their moms had COVID, and they were not sick. So that was actually really important information. We recruited another 47 participants then from 24 states. These are all of our friends and family collaborators across the United States. In the end, we analyzed 316 milk samples collected from 64 women. None of them had the virus. So this really ended that conversation. It was safe to breastfeed a baby if the mom had COVID. And really, they just needed to wear a mask and wash their hands. But we needed good data for that. We've also published studies looking at antibodies, and there's good antibodies in milk. And most recently, we asked the next question, what happens when the woman gets vaccinated? Because that was the next crisis. What happens to milk composition? And we've done multiomics on milk collected here by women who had natural COVID infection, and then the 3 vaccines. We got this milk from Rebecca Powell, who's in New York City. This is just data from the proteomics. You can see when a woman has a natural infection, this is the proteomics of her milk. Lots of changes. When she gets vaccinated, almost no changes. So if you want your milk to stay the same, just don't get COVID. But the virus is not going to change your milk. And here are results from lipidomics and metabolomics where we see the same sort of thing. So a lot of changes with natural infection, but not with vaccination. And we also were able to show that there were no vaccine components in milk, which was also the big concern. So that helped. And finally, our new simple question, just a couple slides on this. We got very interested in cannabis use, right? So we don't know much about the effect of maternal cannabis use on milk composition, but that's a cool question, right? And now that cannabis use is legal in so many states, we started doing this during COVID. We thought, why not? Why don't we just study cannabis while we're home? I don't know. But we got a grant. So we're like, okay, let's do that. So this is really important in the United States. This shows cannabis use in males versus females in different parts of the world. And what's interesting in the United States is we have the highest percentage of women. Okay, of users, 42% are women in the United States. So women are using cannabis. A lot of women are using cannabis during lactation. And you know why? Because they feel like it's safer than using antidepressants and things like that. So they're actually using cannabis for all the right reasons. And we know almost nothing about the impact, if there is any impact on milk composition or maternal and infant health. So we jumped in. We're like, let's do this. This is kind of cool. So we ran a study, a case control, sort of quasi-experimental design study. We recruited 20 breastfeeding women who use cannabis, and I'm talking they use cannabis, not just like once, they use cannabis. And then 20 controls, one of which had cannabinoids in their milk, so we had to kick her out. So she was obviously using cannabis. And basically And so we had the cannabis users abstain for a while, for 12 hours. And then we said, okay, now you can use cannabis, and you can use cannabis as much as you want for the next 12 hours. And we had them collect milk samples so we could see what was going on with the milk. And then we recruited controls and matched them for BMI and some other time postpartum. And we had them collect their milk samples at the same time of day as their cases, because we know that time of day and body fat impact milk composition. So yes. we did complete breast expressions, and our main results that we were interested in at first were delta-9 THC, which is main active cannabinoid in milk. Here's just a picture of what happens to THC after they use. So here's baseline, and then they used whenever they wanted to use. And you can see that this cannabinoid is quite variable, depending on the woman. And some of these women used like 3 or 4 times during that 12-hour period, and some of them only used 1 time. So again, we have a few papers on this, and I would refer you to those papers to kind of parse that out. It's very interesting. Again, we've published quite a few papers on this, so yeah, I would encourage you to look at the literature. We have a very good postdoc in the lab named Irma Castro Navarro. She got very interested in this, and she wrote a review just a couple of years ago. It just got published last year, looking at the theoretical evidence that cannabinoid use might impact milk composition. And again, I would refer you to this review that was published last year if you want to look at it. What was interesting is that from animal studies and cell culture studies, et cetera, it looks like there's really good evidence that cannabis use might impact milk lipids. So then we're like, yes, let's get back into lipids. We know how to do this. And so she actually took the milk samples from that study that I just showed you and analyzed them at baseline and then a couple times after use for the milk macronutrients. These are the data from the baseline period. And you can, with the cannabis group and the control group, and you can see that total lipids, lactose, and protein were not different at baseline. So it looks like chronic cannabis use doesn't impact baseline cannabinoid levels in milk, at least after 12 hours of abstention. However, as hypothesized from the literature, when we looked at milk lipids, and again, we collected complete breast expressions so we could look at lipids. Here we have the milk lipid content at the beginning of the study and then through the 12 hours. The green is the cannabinoid or the cannabis group, and the black is the control group. Now, what you would expect here, because we were collecting milk samples really close together, what happens to milk fat is it goes up if you collect milk samples really close together because you're getting more hind milk than fore milk. And so what you see in the black with the controls is what you would expect. What we see in the cannabis group is they have milk fat depression. So we're kind of back to where we started, looking at milk fat depression and trying to understand what's causing it. So with that, my summary slide, overarching themes, much of which I learned here at Cornell. First of all, the topic of your thesis doesn't matter at all. So that's my thesis at Illinois. I studied selenium nutrition and breastfeeding and babies, never done selenium. Well, now we're doing selenium, and then my PhD, really interesting and cool, control of postpartum fecundity by maternal food intake and suckling stimulus in the laboratory rat. I don't do that at all, not at all. So the topic doesn't matter, but your training matters. Get good training so that you can go out and do all these cool studies afterwards and do them right. The other thing I learned is methods matter. It always matters. Use the best method you can possibly use because it really might differ. So do it right. Third, don't assume what you know is correct. Don't trust dogma. How many times did we hear that, Dale, when we were being trained by you guys? Dogma, like that's dogma. It might not be true. So ask the simple questions first. Don't assume that what you're being told and what people believe is correct. Be fearless, right? Look for serendipitous findings, things that you don't expect to find, because they're usually the most interesting, but they're only interesting if you're curious about them. So be curious. Do meaningful work, It's got to be fun. It's got to, in my opinion, it's got to have some potential impact on us, right? On the world. And then finally, relationships, including your mentors, friends and family, they're really important. Like I said, don't burn bridges. And develop relationships that are long-lasting and that are meaningful. I will say also, I didn't talk about this, but we had 3 kids. Family comes first, always, always, always. Work with good people, be grateful for your mentors, and then finally, try to get away from this idea of independence. What you want is interdependence, where you're taking a leadership role in something, right? You're the leader in this part of it. But working independently, it's not going to solve the wicked problems that we have. So with that, I'll leave you with this great slide. And happy to answer some quick questions. I mean, what questions are there with bagpipers up there? I do have a question about. Any burning questions? Yes.
For the cannabis project,
Yeah.
did you, how do I say this without sounding stupid? Did you provide the cannabis users with samples? Because there's so much variation. Well, I know that's like, I know, that's a big,
Had to go
but like, did you look at the type of strain which they consume, look at the THC quantities in that, because there's so much variation.
There's so much variation, and we had planned to look at like edibles versus inhaled and things like that. It turns out nobody does edibles when they're breastfeeding. They only inhale because it's an easier controlled high. So so no, we could not do anything like that. The IRB was very specific, like you just let them do what they want to do and don't intervene, and you just see what happens.
Okay.
So no, but it's a good question.
Yeah.
Yeah.
I have a question. In one of the first papers that you talked about, where you sent your breast milk to a national,
Yeah, the Monsanto study. Yeah.
or no, I think it was before that, where you sent
microbiome
these
study.
samples to an external company, and they said, okay, all of these samples are sterile.
Yeah.
What was their explanation for why those samples were sterile when you saw that?
Yeah. So they were doing culture dependent. They're using like 10 microliters of milk. They're only looking at specific pathogenic bacteria and using that method. I mean, 10 microliters of milk isn't a whole lot, and milk microbiome is low biomass, and they were only looking for specific pathogens. So it was like the perfect storm of nothing is there.
Lab designed for dairy.
Lab designed for dairy, mastitis. Yeah. If we hadn't seen it with our own eyes, we wouldn't have questioned it.
Just a quick follow-up, and thanks a lot, Shelley, for the presentation. It was great. So where do the bacteria come from specifically? And, where do they reside in the tissue? I have my cow bias, so, I wanna.
I mean, so it turns out there are bacteria in the mammary gland long before breastfeeding. We look at biopsies of mammary tissue of, women who have never breastfed, there's a microbiome there. So it's there early, it's persistent. We think I don't know where it's originating, but there's a lot of evidence, at least in breastfeeding women, that it's coming from GI tract via enteromammary pathway, probably via the lymphatic system, trafficking to the mammary gland. And then also we're almost 100% sure it's coming from the infant's mouth. So every time a baby suckles, there's this backwash and it goes back and forth. So I think coming from both of those, but where it's coming from to begin with, I don't. Yeah, job security, if anybody wants like a career path, study the milk microbiome, because there's a lot to learn.
Other questions? Thanks, Shelley, again.
Okay.
To Dr. McGuire. And it's been great having her here, and it's been great for me to get back to Cornell as well. Thank you all for coming. Shelley, thank you.
Thank you.